Okay, Bio30s. I just had a meet and got a chance to talk with a couple students and go through last week's assignment. Just a couple of clarifications to talk to you guys about. So from our reproduction summative, when we were looking at question 2 here, it was asking about this MESA procedure. And so again, when you get something like that, you go back and you look up in the context and see, what are they talking about this MESA business? It's not the place in Phoenix. So then you read, oh, MESA, that is microsurgical epididymal sperm aspiration. Epididymal, well, that must be the epididymis. So they're saying this procedure is used to obtain sperm. So they're sticking a needle, sorry guys, I know you're all wincing now, right into that epididymis. And then they take that sperm and it's used for in vitro. So they combine it in a dish with some eggs from a female. And then where are they going to put it? Well, they're going to put it right into here, right into the uterus, number 9. Okay, so that is one question that a lot of students, more students actually got the wrong answer on that one than the correct answer. So structure 5, the epididymis, they're not taking the sperm directly from the testes. They're going up into the epididymis to obtain the sperm. Okay, another question that students got wrong, and in chatting today with a couple of people, it seems like maybe people were freaked out by the fact that this answer was 4231. So the correct answer actually was 4231. Sorry, 4321. And I wonder if just, answers like 1, 2, 3, 4 or 4, 3, 2, 1 freak people out. Don't let it. I mean, it's a totally viable option, and in this case it was, right? And so if you look at this, in the first week is mitosis, right? Cleavage. And then implantation is next. The embryo buries into the endometrium. Then in the third week, differentiation, we're getting our germ layers, mesoderm and ectoderm and endoderm. And then after that, there's going to be a beating heart. So 4321 was the answer, and I don't want those answers to scare you, okay? All right, next question. A lot of students, I don't know if maybe you were mixing up the, oops, sorry, my bad there. If you were mixing up the uterus and the amniotic fluid, because a lot of students, for this question, they did 4, 2, 1, 3. Others had 1, 2, 4, 3. I don't know. This one just seemed to be very poorly done, and a lot of students did not get it correct. So just to do a quick rundown, that the uterus is the mom's organ that stretches and expands, and it houses the developing fetus for 9 months. The placenta developed from the chorion. Remember, that's where the chorionic villi were, and that's where the gas exchange and nutrient exchange is occurring. The umbilical cord is this little tube that is going to take exchange gases and nutrients to the fetus, but remove waste to the placenta for exchange. And then lastly, the amnion. Now the amnion is this layer here, and it produces a fluid. So all this stuff around the fetus, that's the amniotic fluid, and that's a shock absorber. It protects against pathogens, etc. Okay, so those are some common errors from that summative. Now, from our formative, a lot of students were confused by, there's this terminology, right? Chromosome versus chromatin versus chromatid. Now, I know that is a lot of words that sound the same. And so remember that chromosomes are these condensed genetic structures that we would see here. Like this cell number 1 is in metaphase. How do I know that? Well, all those chromosomes are lined up on the metaphase plate or the equatorial plate. So that's what a chromosome looks like, that little X-shaped structure. Now, when in anaphase, the chromosome separates and becomes 2 chromatids, and then they go to the opposite poles. This little half a chromosome guy, that's a chromatid. And then finally, this long stringy stuff that we see here, this is telophase. We're just about to go into cytokinesis. These two cells are dividing into two. And also we see that in prophase, where the chromosomes are just starting to form. But this long stringy stuff that just looks like that in the nucleus, we call that chromatin. And that chromatin has not yet formed into a chromosome. Okay, so several students were confused by that terminology and these stages. Another confusion, there were some diagrams of cell division, and some students got a little confused on this question. So we see here's cell A, and then it has one division. This process must be mitosis because there's only one set of divisions, not two. And we're producing body cells, right? Exact copies. Obviously, we only get one of the two chromatids, right? Because this one will have gone to the other pole. And so this is just identical copies, right? This is what happens in your body cells. This is not a zygote. A lot of students seem to think this was a zygote. Well, it has too much genetic information for there to be a zygote, so it is not a zygote. Okay, and then another question that students had trouble with was the question where they were saying that they were trying to draw metaphase 1, but it wasn't. This is not metaphase 1. And how do I know that? I look at the chromosomes. A chromosome is going to be found in meiosis 2. Right? When we're talking about metaphase 1, prophase 1, we're talking homologous chromosomes. We're talking double the amount of DNA, right? A pair that matches for size and shape. So I'm talking about a tetrad. That's what we should have been seeing in this diagram, not a single chromosome, but rather a tetrad. So here's an example showing this is what it should have looked like in their diagram, right? This is what we call a tetrad for whatever chromosome that is, number 1 or number 21, whatever. That is what we call a tetrad. So they should not have been drawing chromosomes, but rather paired or homologous chromosomes. And the orientation doesn't matter, right? You can have your equator going this way in the cell or your equator going that way in the cell, but either way, when we're in metaphase 1, we're talking about homologous pairs of chromosomes. So that was one misconception that students did not understand. And that event in metaphase 1 that causes genetic diversity in metaphase 1 is called independent assortment. It is not crossing over. That happened in the previous step in prophase 1. So again, there's two events in meiosis 1 that is going to ensure diversity. The first one is happening in prophase 1, and so that was the last question. Prophase 1 of meiosis is where crossing over occurs. Metaphase 1 is where independent assortment occurs. So this week we've got several video lessons on how to do genetics questions, and there's a lot more terminology to learn. There will be 2 assignments this week, a formative on genetics and a summative on cell division. So you guys remember, if you are stuck and if you're having questions and you're working through the work on genetics and you're like, oh, I need some help, well, email me, right? michelle.ledeen@ghsd75.ca. Okay, so that's it for now. And again, I'm no further away than an email. So take care of yourselves, and until our next Google Meet, talk later.